So welcome everyone on behalf of Precision NanoSystems to today's webinar on developing RNA medicines for the treatment of pandemics. I hope everyone is safe and well wherever you are joining us from. So we're going to be talking about RNA medicines, and especially RNA-based vaccines are attracting a lot of attention as an attractive modality to tackle COVID-19. So we've seen news reports and data coming out of many organizations such as Moderna, CureVac, BioNTech, with Pfizer, who are advancing RNA vaccines for COVID-19. So at P&I, we're also pursuing this strategy. So today we thought we wanted to explore with you why RNA vaccines and also therapeutics are particularly attractive when we need to respond rapidly to a crisis. So we'll be focusing today on RNA lipid nanoparticles, which have been clinically validated, at least as a therapeutic, with the recent approval of Alnylam's siRNA lipid nanoparticle drug Onpattro, which is approved for the treatment of a rare liver disease. So today we're looking to learn from our panelists about their previous experience in applying RNA medicines in a pandemic. We'll also explore the differences between RNA lipid nanoparticles developed as a vaccine versus as a therapeutic. We'll discuss manufacturing of these particles to ensure supply of medicines to those who need it most. And we'll also focus on key considerations in being prepared to rapidly respond to a pandemic such as COVID-19 and be prepared for any future pandemics. So what I would stress here, though, is while we are framing the discussion around response to pandemics, the information we will cover will be applicable to RNA medicines used to treat and developed for other conditions where there's an unmet medical need. So again, warm welcome to everyone participating here. My name is Euan Ramsay. I'm a co-founder and I'm the Chief Commercial Officer here at Precision NanoSystems. Today, the webinar will be a panel format. I'll be your moderator, and to kick off the discussion, I have some questions for the panelists who will introduce momentarily. So let me introduce our panelists here with me, my colleagues. First of all, I have Dr. Andy Geall. So Andy has recently joined Precision as the Chief Scientific Officer. Andy joins us from San Diego. In a previous role, he led the RNA nanoparticle vaccine platform at Novartis and was the principal investigator on that DARPA contract, which was applying technology towards pandemic response. So Novartis and collaborators demonstrated proof of concept for development of a fully synthetic H7N9 flu vaccine in less than 2 weeks after sequencing of the virus. So very pertinent for today's discussion. Most recently, Dr. Geall was an executive at Avidity Biosciences, which has established an antibody oligonucleotide conjugate platform, particularly focused in treatment of rare muscle diseases. So welcome, Andy. Looking forward to your insight. And then also my colleague, Lloyds, based here in Vancouver with myself. He's been with us for 2 years. His role is the Director of Clinical Manufacturing Solutions, and Lloyd brings over 20 years of experience in the industry, particularly working on lipid-based drug delivery systems. So Lloyd, PhD in applied microbiology from the University of Saskatchewan, previously worked at Northern Lipids and Arbutus Biopharma. So Lloyd has expertise in formulation and process development, and he's played key roles in technology transfer and GMP manufacture of RNA-based lipid nanoparticles. In that case, he supported multiple clinical stage product candidates, and then again, pertinent to this discussion, including an RNA-based Ebola drug that was developed for the US Army. So welcome, Lloyd. Okay, so let's get started here. So I think as we mentioned in your introductions, you've obviously both got previous experience tackling the pandemics here. I think maybe, Andy, we will start with you, because
Sure.
you were working with Novartis at the time, and this was early on, I would say, in the development of mRNA-based vaccines, and there was an H7N9 avian flu outbreak. So maybe you can describe, provide us the story behind that.
Yeah, I mean, the first thing I'll point out is that this wasn't a pandemic. There was no human-to-human spread. This is an H7N9 influenza virus that jumped from birds into humans, and there were 3 case fatalities. And the Chinese CDC sequenced the viruses and put the sequences online, available, to the world. And so at the time, this is 2013, towards the end of March, and Novartis Vaccines had just constructed a mammalian cell flu culture facility in Holly Springs and was looking to speed up RNA production for traditional systems. And to grow virus in cells, in mammalian cells, you need a seed virus. And traditionally, that virus isolate has to come from the site of infection, its passage through eggs, it mutates, and then once it's viable, you use that as your seed stock in fermentation. So Novartis was looking to accelerate that process and was working with synthetic genomics, and they were synthesizing just the outside of the virus, the HA and the NA genes. And flu is a segmented virus. It has 8 segments, and at a high level, 6 of those segments are for the production of the virus, 2 of them are for the outside. So you can reassort 2 viruses and get the coat on the outside that you want, and it's viable to grow. And so that reassortment process was being developed. We decided to circumvent the reassortment process and just take the sequence. And so the day that sequence came out, I got a phone call from the head of research of Novartis Vaccines, Rino Rapoli, and he said, look, if we can get you the gene, how quickly could you make a vaccine? And how quickly could you demonstrate that, the speed of the technology? And so we just agreed to do it. It was a Friday afternoon. The guys came in the lab over the weekend to get things ready. And 3 days later, the synthetic HA, the H7, arrived from Synthetic Genomics in San Diego by FedEx. They cloned it into the DNA vector. They ran the transcription reaction, purified the RNA. We threw it on some cells, showed it expressed the antigen on the cell surface. Fortunately, we had an antibody that cross-reacted from a previous isolate, an H7N3. And we then lined up a mouse study, put it in mice. Day 13, it went into the mice, and we showed that the vaccine worked and was capable of raising a neutralizing antibody titer. And then we went ahead and published those data. That was in Emerging Microbes and Infections. The lead author on that, the first author was Armand Heckley, who was the lead synthetic biologist who built the constructs. So that was the first demonstration on an RNA format that you could generate immune response and a viable vaccine. There, we were showcasing our lipid nanoparticle technology, and in the labs, we were, using the benchtop nanoassembler to put those formulations together.
Yeah.
So hopefully, it's not brief, but a detailed, introduction to how we showcased RNA as a vaccine technology.
And so, in that instance, Andy, you were kind of showing proof of principle and that rapid ability to adapt your platform to meet particular needs.
And now we're seeing, RNA technologies at the forefront of the COVID-19 pandemic response. But people have to understand there's 3 different types of RNA vaccines. You have base modified RNA. This is the approach that Moderna is taking, where you introduce mammalian base modifications to dampen down the immune response to the RNA and increase protein expression. Again, they're delivering with a lipid nanoparticle. Then you have traditional mRNA with wild type systems. This is CureVac, and they're again delivering with a lipid nanoparticle, pushing very fast to go into the clinic. The third type is a self-amplifying RNA. This is based on the genome of an alpha virus. This is the work we pioneered at Novartis. Again, as we showed in that paper, in a lipid nanoparticle. And we'll see Robin Shattuck at Imperial College pushing that technology with a lipid nanoparticle into human clinical trials. So from a technology base, it's going to be interesting to see how these three different types of RNA vaccines with a common theme, the same delivery system, perform in clinical trials. Obviously, the lipid delivery systems are different, the vectors are different, the antigen design may be different, but from a broad-based approach, we should get an idea how well these vaccines perform. Producing RNA is a generic process. It's the same raw materials, but the dose is going to be very important. The lower the dose, the smaller the manufacturing footprint, the more capacity you have. And in a pandemic, it's all about capacity and how many vaccine doses can you produce. Yep. And probably the literature would suggest self-amplifying RNA has probably the best chance of doing that. We'll find out. Yep. You know, predictions are about a microgram for self-amplifying RNA, a base modified with Moderna. From reading the literature on their clinical trial designs, somewhere from 25 to 200 micrograms, we'll have to see.
Yeah. Well, that's great, Andy. You've covered a lot of information there, which we'll delve into deeper in a minute. And I think I'd like to bring Lloyd in, because your experience is a bit different here, Lloyd, because you were working with a therapeutic, an siRNA lipid nanoparticle, if I'm not mistaken, and you were working with a technology which was clinically validated. So maybe to the extent you can do, we'd love to hear more about your experience.
Yeah, in general terms, my former colleagues at Arbutus, and formerly known as TechMira, before that Protiva Biotherapeutics, developed a LMP platform technology to be used as a therapeutic platform for delivering RNA-based drugs. So that work had involved many people, too many to list in this call. It'd probably take the next hour listing all the people involved. So it was a real team effort. And also, I think when you get an outbreak or a pandemic, I think it kind of underlines the fact that, are you ready to go? So, you know, years of work, also a contract with the US Department of Defense in 2010, really put us in a pretty good position to respond to the outbreak in West Africa that started in 2013, I think peaked in 2014 and 2015, to allow us to take the drug substance, in this case, the RNA interference drug, or the siRNA, to tailor it specifically against the disease, the virus disease in this case. So we could move quickly, and other companies are demonstrating this too, because they have the infrastructure in place, they have the delivery vehicle, and they know how to make it, they know how to test its efficacy, and know how to test its activity in preclinical studies. So it's sort of clinic ready for the clinic and ready for experimental use. I think it's important to realize that the use of experimental therapeutics is probably the first thing that happens during an outbreak, and also the use of drugs that have been approved for other purposes, have been repurposed. And we're seeing that today with the COVID-19 response. And to be honest, you know, the more people working on this, the better it is for the entire world. The more minds working together, and hopefully collaborating together as well, sharing data. You know, hopefully we can find initially effective treatments, and that needs to be demonstrated through clinical trials. And again, the long-term plan, or hopefully not so long-term with the emerging technologies, is to find an effective vaccine. Because I think, to Andy's point, he was talking about microgram doses, typically with intramuscular injection, whereas therapeutics using nanoparticles are multiple doses, 5, 6, 7 daily doses at much higher concentrations. So, you know, up to probably 500 or 3 orders of magnitude higher doses for a therapeutic than you need for a vaccine. So manufacturing becomes more of an issue for a treatment versus a vaccine. So I guess, was it Thomas Edison? I think he said that an ounce of prevention's worth a pound of treatment. So we need to think about preventing this long term, and I think viruses, vaccines to these dangerous viruses are very important. Also, viruses change, as we know. So for the future, I think we need to keep our eyes open, and we saw that for the Ebola situation, where therapeutics have been developed for a certain kind of Ebola virus, and the virus in West Africa outbreak it was different. So we had to fine-tune the payload. So that's important to keep in mind. So if you have a platform technology, you can make those minor adjustments quite quickly, do the necessary safety studies and the chemistry and manufacturing control adjustments as well, and then respond quite quickly. As Andy mentioned, we can manufacture these synthetic therapeutics quite quickly if you have all the raw materials and you have the manufacturing processes, and those specifications for the raw materials have been validated, and you also have a qualified manufacturing process where you will keep process parameters in your critical process controls.
Yeah, so I think you covered a lot of interesting points here, and I think we talk about this concept of a platform and the ability to modify and adapt it to respond rapidly. And I think RNA in itself is a platform. The lipid nanoparticles are a platform, the manufacturing is a platform. But I think you touched on this, Lloyd. The lipid nanoparticles originally developed for siRNA, so it's administered intravenously at large doses, is designed for multiple dose formats. But Andy, that's not the same administration route, dosing considerations for a vaccine. So maybe you can, in the spirit of like a target product profile and thinking about the product form, maybe you can expand on what the critical characteristics of a RNA LMP, if you're developing it for a vaccine as opposed to a therapeutic.
Yeah, so for the self-amplifying RNA systems that we published on at Novartis, and it's pretty common target product profile across the board. So we're looking at a traditional intramuscular injection with a standard needle and syringe. Volume is usually 0.5 ml, and after administration, you want that vaccine to stay at the site of injection, or drain to the draining lymph node. You'll get expression of the antigen in the muscle cells at the site of injection, and then you'll get some immune stimulation from that RNA, both through delivery of the RNA to the immune compartment, and also the amplification of the RNA in the transfected cells and expression of the antigen. Right. Doses are typically a microgram. So Lloyd is talking about delivering an siRNA to knock out a gene of the Ebola virus to stop it replicating. There, they typically dose in, let's say the dose, it's somewhere between 0.5 to 1 milligram per kilogram. A typical patient would be, say, 90 kilos. So you're looking at dosing 90 milligrams of siRNA as opposed to 1 microgram of self-amplifying RNA. So very different concepts. Obviously, the therapeutic effect is immediate within hours. For a vaccine, you typically, if the patient's naive, you're going to need a couple of doses, a prime and a boost. For a self-amplifying RNA, that interval will be somewhere between 5 and 8 weeks. And then you need to allow time for the immune system to generate a potent response. So it's going to be months before you see the benefit, the prophylactic benefit of that vaccine, as opposed to the siRNA, which is immediate. So if somebody has an acute infection, the therapeutic can be used. The vaccine is really there to healthy people around that hot zone of infection. You try and ring vaccinate and prevent spread out from the containment zone.
Okay, and so I think, again, an important point you brought up there, Andy, the difference between a vaccine and a therapeutic in this case is that we are looking for an immune response with a vaccine.
Yes.
And so in that way, the considered liabilities, if you like, sometimes the lipid nanoparticles actually are beneficial in this instance. Is that true, or how important is the immune response from the carrier in this instance?
Well, I mean, so there are three essential components to an RNA vaccine, and you've got to get all three right. So you've got to design your antigen correctly. And so for COVID-19, they're going in with the spike protein. And, you know, the literature is quite clear. If you get it wrong and you don't raise the right kind of immune response, high neutralizing antibody titer, you could get antibody-dependent enhancement of disease. So you've got to get that right. You've got to get your vector right, because it's got to be able to express the protein in the right place. And then the delivery system does matter too, because it's not only got to be capable of, for the self-amplifying RNA format, delivering to the muscle cells, transfecting them, allowing that RNA to express the protein. It's also got to engage the immune compartment. And yeah, not all lipids are created equally, and that adjuvant response will be different dependent on the type of LMP that you have.
Okay, great. So I want to quickly move on to just get a brief question in on the manufacturing considerations, and then really I'd like to open it up to the participants here to get some of their questions to you, gentlemen. So Lloyd, maybe just walk us through, you've obviously got quite a bit of experience in the GMP considerations for manufacturing these RNA LMP, so perhaps you can give us an overview on considerations when we're making the move from preclinical to clinical, and then maybe I can also get you to comment on is there any special considerations with respect to dealing with a pandemic, particularly if there's a localized outbreak?
Yeah, so hopefully everyone can hear me. I'm going to speak a little bit louder for everyone's benefit. So if you, Euan or Andy, you just let me know if I need to speak even louder than I am. Importantly, yeah, so we're talking about preclinical R&D work with a promising, say, therapeutic or a vaccine, making the move, making plans to make material that's going to go in human beings, right? Making a drug that's going to go into human beings, or a vaccine that's going to human beings. So it's pretty clear you've got to go through, the regular, be ready to prove to regulatory authorities that your drug is safe and at least preclinically effective. So to do that, from a manufacturing point of view, we use good manufacturing practices, or GMPs. And everything that goes into the drug is traceable and is basically tested before it's used to manufacture your drug. So for a lipid nanoparticle, the input materials are lipids, your final buffer, your payload, which could be a nucleic acid of different kinds. It could be an siRNA, it could be a messenger RNA, or a self-amplifying RNA, and there's others as well. And you have to source those and ensure that those are manufactured according to good manufacturing practices. So there's full traceability on the starting materials. It's auditable. You can basically reduce any chance that there's a contaminant in one of your starting materials. And when you're going into actually manufacturing the lipid nanoparticle batch, you have that documentation in place. So for manufacturing, you need to have a plan. You need to have a batch record, and that batch record needs to be tested. It needs to be basically test run with at smaller scale and sometimes at scale, to ensure that when you make the material that's going to go to human beings, you've worked out all the kinks. You've worked out any kind of potential issues and what have you. So that provides a little bit of a challenge when you're developing a process on the fly, where you do not have the raw materials on site or in place, and you have to basically ask your vendors and beg sometimes to provide the materials that you need for this urgent need. So in an ideal world, those materials are, you have those on hand, or they're readily available, and you have them at your manufacturing site. So another important consideration is that you're developing a preclinical process. You've locked your formulation, you know what your lipids are, you know what your payload is, you've you've probably done some screening exercises to find the best mRNA sequence. You've maybe looked at chemical modifications. You have preclinical, in vitro or in vivo, good idea that it's the most efficacious, potentially the safest of the selection of payloads that you're looking at. And then you have to really, you've locked down your formulation, you've locked down your process, as I've mentioned, you know how to make the drug, you know every step of the way, you know how you're going to fill it, you know how you're going to store it, you have an idea how you're going to ship it to where it's needed. And you perform basically at that point, you go to a GMP manufacturing facility. The larger companies, they have those themselves. There are a few government-run facilities. And also, obviously, there's contract manufacturers and CDMOs, contract development manufacturing organizations as well. So the tech transfer of your process, your formulation, and how you're going to make your drug product is very important. And that takes time. So again, pre-existing relationships, having that established ahead of time is really going to speed up your response. So I think there's a ton of work that goes into developing a drug that's ready for clinical trials. And for a normal therapeutic, that can take many years. So in the case of a pandemic or a rapid response, the hope is that though that framework is already in place, you already have what you need. So the changes you need to make are relatively minor. And things you can focus on, the most important things you need to do. And typically that's going to be manufacturing your payload, your drug substance, which is specific for the virus infection.
Okay, thanks, Lloyd. And Andy, when we've got some questions coming in here, but just to finish, maybe close off this manufacturing section, then from a vaccine perspective, what do you see as the ideal manufacturing format? Is it a global, we make it in centrally, we ship it? Is it in country? Is it, in a back of a truck, can we drive it to the site? How do you view delivering the actual product to the patients that need it?
I mean, I think, the current situation is clearly indicating that, people have to have in-country manufacturing. Now, obviously, the smaller that manufacturing footprint is and the more automated it is, that you can then, rather than just having one facility in country, you can start to have, production at sites like hospitals, and really reduce the supply chain, the need to distribute the vaccine out. We're not there yet. I think we've arrived at the point, certainly the global capacity for mRNA production isn't there to supply the current outbreak. I don't think we even know that these RNA vaccines work. We'll find out. The solution will be a combination of probably 5 or 7 different vaccines to cover the global population. But I think what we can learn from here is, that distribution is important. We'll go to in-country, and then we're going to see further innovations on RNA technology to bring down that manufacturing footprint, automate the process, and you'll start to see it implemented even in a local setting within each country.
Yeah, so ideally we're getting to this point where we recognize that we have to have the building blocks in place, and if we can have a manufacturing, I know Andy and myself are both pharmacists, and I think we're of the kind of view that we've worked in hospitals, we've produced drug products in hospital, and we could imagine that you could use existing infrastructure to deliver these drugs at some point in the future.
I worked in an aseptic dispensing unit, in a hospital pharmacy, and made products that were then released, went up onto the ward, and were dosed into patients. So, you just need the technology, regulatory alignment on how to release those vaccines, and that will take several years. But the science is definitely there, and now it's a question of building on the science, working with the regulators, working with governments on how to build the infrastructure to do this differently next time. Yeah. The science and the technology is definitely there.
And then I think we're contemplating, rather than stockpiling a given vaccine, given that who knows what the future pandemic would be or how COVID may mutate. What we're really looking at is getting a regulatory basis for the raw materials if we want.
Yeah, you stockpile. Stockpile the raw materials.
Yeah.
The RNA bases are always the same. We're just emailing an electronic sequence now of that, the key antigen. But I will stress, you need to get that antigen design right. And you've got the three components: antigen design, vector, delivery. And you need all three. And then the fourth is you need a manufacturing process that's robust and capable of supplying that vaccine in the appropriate quality.
Yeah, great. And this is great. So now I've got some questions. I appreciate all the questions coming in from everyone. And this is, I think, a good segue here, because there's a lot of questions around the mRNA and the different, approaches. So what's BioNTech doing? What's Moderna doing? What's CureVac doing? What's Robin at Imperial College London doing? So maybe you can speak a little bit more about the RNA, the messenger RNA itself, Andy.
Yeah, I mean, so there's three different types. There's two short RNAs, two conventional RNAs. One is base modified, one is unmodified. So Moderna's focused on the base modified systems. And so this was, the first incorporations of mammalian RNA-based modifications into mRNA was pioneered at the University of Pennsylvania by Katie Carrico and Drew Wiseman. And they showed that if you incorporated those in, you could make them non-immunogenic. The human immune system would see them as self. And so that's the approach Moderna is using. And it's a different technology to the technology that CureVac's using, where they're using wild-type RNA. It will have an adjuvant response, but it's also capable of protein expression. And so they focused on putting elements in the 3' and 5' end to try and increase the level of protein expression, the antigen, and they focused on trying to get effective delivery. The third type, these are much larger self-amplifying RNAs, through 2 to 3 kb for Moderna and CureVac. Self-amplifying RNA, 10,000 bases, depending on how big the antigen is. So a little harder to make, but it's self-amplifying. So if you get it in the cell, 1 copy will become 10,000 copies, and the level of expression is much higher. And obviously that RNA is a viral genome, and your immune system will see it as that, and we're hot-wired to react to our immune compartment seeing a viral genome. So 3 different technologies. I know there was a question from somebody, what are BioNTech using? Nobody knows. They have all 3 technologies within their portfolio. And so I don't think we know what they're proposing to take forward at this time. I'd say we don't really know the details of what CureVac are doing, Moderna, Imperial College. I'm extrapolating from papers that they've published on what they're doing. But it's clear we have 3 different approaches and technologies, but it would appear that everybody's focusing in on that lipid nanoparticle as a key delivery technology.
And there's a question on this, Andy, then, it relates to these diverse RNA modalities and also the LMP choice and combination. And so how do they influence cellular versus humoral responses? And the question here is, why aren't more intracellular viral epitopes being selected to drive durable T-cell responses?
Okay, so the preclinical data on T-cell responses for the 2 conventional technologies aren't that great. Certainly better with a wild-type RNA approach. For the self-amplifying RNA, which is my area of expertise, you see great T-cell responses. We've published 20 papers in the field. If the people asking these questions want to know, the data's already there. The first paper we published was in 2012 in PNAS, and we focused on respiratory syncytial virus. Buried in the back of the supplemental data is all the T-cell results. Commercially, vaccines tend to be licensed based on antibody responses, and neutralizing antibodies, and that tends to be the focus. But there's no mistaking that a self-amplifying RNA has both an antibody and a T-cell response, and this might be important for, you know, a coronavirus. And the type of immune response you get for a self-amplifying RNA, it's a Th1 response. It's probably what you're looking for a coronavirus, to avoid those unwanted disease enhancement that you might see, because the type of antibody you express to neutralize the virus has you know, Fc domains that can activate the immune compartment. So certainly we see for self-amplifying RNA robust T-cell responses, and we're seeing implementation of self-amplifying RNA in the cancer vaccine space, where those T-cell responses will be critical for clearing tumor.
Okay, and then, sorry Lloyd, I'll definitely get you a question here, but another one here for Andy, and I think this kind of relates to your role at Novartis Vaccines. So the question really here is, why are lipid nanoparticles the predominant carrier for these mRNA vaccines versus polymers or other approaches? So at Novartis, clearly you were relatively agnostic in the carrier. So how did you?
Yeah, I mean, at Novartis, we developed two delivery systems: lipid nanoparticle and a cationic nanoemulsion. The cationic nanoemulsion is it's a squalene oil emulsion. It was Novartis to develop the adjuvant MF59 for protein-based systems, and what we did is we added a cationic lipid DOTAP to that system, and it was a very stable nanoemulsion, and we adsorbed the RNA to the outside, and it was simple to produce. It was a two-component system. You mix the RNA at the bedside and you vaccinate, and it was pretty robust. And if you look at the preclinical data, we were seeing immune responses in the 10 to 50 micrograms. The LMP systems were much more potent, but very difficult to convince the pharmaceutical organization that it was a scalable technology at the time. We haven't, you know, the advances that have been made by Precision NanoSystems on microfluidic mixing is phenomenal. You've got GMP systems now capable of producing large volumes. They didn't exist there, so there was a lot of nervousness around using an LMP, but preclinical data is really clear, very potent delivery system for an mRNA vaccine. And we've seen Moderna using it and publishing it. They're less conservative, and you know, biotechs tend to push the envelope a lot. And we've seen CureVac publish and switch away from their protamine now to an LMP delivery system.
Yeah, okay. I think it's definitely worth mentioning that all of this is obviously built off of the clinical validation that was originally done for the siRNA. So it's certainly been de-risked to that degree. But Lloyd, what about this question here? This relates to the choice of lipids, the lipid components within your liposome or your lipid nanoparticle. Now, people are asking the question, what's the role of the lipids on the best choice of lipids, I should say, for the route of administration? So if I was administering it intravenously versus I wanted to administer the drug through into the lungs, pulmonary, can you comment on the lipids or the lipid formulation characteristics you might look to optimize for those different routes?
Definitely, yeah. So there's been quite a lot published on kind of the base components of lipid nanoparticles. They all have 3 or 4 components in common. For nucleic acid lipid nanoparticles, the cationic lipid is what helps the negatively charged nucleic acid become encapsulated within the lipid nanoparticle. Those lipid nanoparticles are typically around 100 nanometers in diameter. They can be a little bit smaller, they can be a little bit larger. For treatment, most of those treatments were systemic. So after like an IV injection, the lipid nanoparticle drug containing your payload needs to circulate in your bloodstream until it gets to its site. Usually that site is the liver, although nanoparticles, lipid nanoparticles, can be designed to go other places as well. For a vaccine, I think the majority of vaccines, Andy can jump in if I misspeak, are administered by intramuscularly or locally. So there's no requirement or less of a requirement to have extended circulation in the bloodstream, and that would inform on the design of the lipid nanoparticle.
Yeah, and I think there's obviously, when we talk about intravenous, it really depends on where you want your nanoparticle to go. And I know there was a question actually sent earlier ahead of the panel here. I was asking the question about apolipoprotein E and its influence in the uptake of lipid nanoparticles. We certainly know that to the liver. Lloyd, maybe you want to comment a bit on that.
Yeah, I think there's a number of papers on that. I think P&I even has a video of that as well on our website that people can have a look at. But yeah, there is a kind of, you have to understand, once you inject your lipid nanoparticle into the bloodstream, or any site for that matter, there will be some interaction with the hosts, whether that's an immune reaction, whether that's interacting with circulating proteins in the bloodstream. And that comes basically into the design of your lipid nanoparticle as well. You have to understand that to basically develop or design an effective delivery vehicle.
Yeah. And maybe, and I just want to, sorry, go ahead, Andy.
So Euan, when it comes to, ApoE and delivery mechanisms for vaccines, for local administration, there really isn't that depth of knowledge yet. I certainly, when I put up a slide of how the LMP, delivers to a myocyte or muscle cell, I have, it either goes in through direct fusion or it goes in through endocytosis. Is ApoE involved? Probably not, but the science isn't as in-depth as it is for systemic delivery, where they've spent a lot of time and effort to fully characterize the delivery mechanisms.
I mean, I think this is probably another subject for another panel, but certainly depending on your route of administration, your target cells, size, charge, components, all these have a big influence on the the activity, the relative stability of a particle as well will have a big influence. But let's get on to there's lots of questions here around cost here. So how does the cost of RNA and lipid nanoparticle manufacture play into its potential to be used widely? So maybe Lloyd, you've got the experience of working through the last 20 years in this field, so maybe you can talk about the advances they've had and what influence that has had on maybe cost and the time taken to manufacture these systems.
Definitely, yeah. So there are quite a number of GMP lipid manufacturers, and there are a number of off-the-shelf lipids for your lipid nanoparticles. A lot of great GMP lipid manufacturers, and I'm not going to list them all here, but you know who you are. And maybe after the call, if you have specific questions, I can kind of put you in touch with them. The cost of lipids actually are a significant portion of the cost of goods for making a lipid nanoparticle. However, the largest cost is going to be your drug substance, or traditionally has been, whether it's an siRNA or a messenger RNA. And anybody working in the field, if they're buying, say, R&D grade messenger RNA for their preclinical experiments, appreciates that cost. The costs are coming down, which is good news, as more people adopt the technology. And in the case of self-amplifying RNAs, as Andy's pointed out, you need a lot less. So driving your cost of goods is the amount of drug substance. And if you have a self-amplifying RNA and you can dose very low doses for a vaccine application, the amount of material you need is quite modest and can be made quite quickly at low cost. The fixed cost will be the regulatory oversight, your GMPs, documentation, quality control, quality insurance, and control of your supply chain, so vendor qualification activities, etc. So the good news is the cost is coming down for these specialty RNA payloads, but also the effectiveness of these LMP-based treatments is increasing as well. It's increased exponentially probably in the last 10, 15 years. So that's due to scientific advancements. So I know we're talking about making clinical grade lipids, clinical grade payloads to make vaccines and therapeutics for important unmet medical needs, but the basic science has to be there, and that's happening in companies, in academic labs, and also in government labs. So again, it's a collaborative effort, and I think the more sharing there is between these groups, the faster we can move. I think one of the questions we might get is, can you recommend a lipid nanoparticle formulation for us to use? I think there are some published formulations, but at the same time, I don't know the components or the composition of the formulations that are going into the clinic today that are being used for COVID-19. So that's one of the things. They're not disclosed, and there's limited details at this point. Hopefully in the future that information will be more widely disseminated and shared. So I think a lot of developers are developing their own formulations. So that is a potential bottleneck and will slow things down.
Yeah. I mean, certainly this is the raison d'etre of Precision NanoSystems, was to look at ways that we could accelerate the manufacturing of these systems from preclinical to clinical, but that's enough of my silly sell. So Andy, what about taking and building on that? This is one for you. There's a question about production costs and length of time for manufacturing these synthetic LMP-based vaccines versus a traditional inactivated virus vaccine. How would you compare and contrast the development costs of that?
Yeah, I mean, there's no cell culture involved here, so you don't have that 2,000-liter stainless steel fermenter and the downstream purification systems to get rid of cell debris, and that takes time to do all of that. You have an enzymatic transcription reaction, potentially all in disposable equipment, and a typical transcription reaction runs for a couple of hours. Now, obviously under GMP, you've got setup times, but you're talking, weeks rather than months in terms of, producing the material, formulating the LMP as well. For self-amplifying RNA, the footprint is tiny. We published a paper out of Novartis, Jeff Ulmer and I, in 2015, where we reviewed what the manufacturing landscape might look like based on a 1 microgram dose. And so from a 1 liter transcription reaction, we showed that if the human dose of the vaccine was 1 microgram, we could produce 5 million doses. So that's 1 liter. So it's not a huge scale process for a self-amplifying RNA, and so it's ideal for a pandemic situation. And really the cost, comes down to the enzymes needed for that transcription reaction. Those costs are coming down, but the biggest cost is the release of the vaccine. So all of the analytics you need to do, and typically, you can put a ballpark cost around $1 million, $1.5 million. So in terms of, it's very much more rapid in terms of weeks of a process. And in terms of cost, if you're producing 5 million doses, it's hard to have the fill finish capacity for that. But you can see that the cost is very small, and the time in the manufacturing suites to produce it is reduced as well. As RNA becomes more mainstream, the cost of the more raw materials will come down too. We'll start to see automation of the RNA process, the LMP process, and it will get shorter and shorter in terms of production time.
Okay, great. And so there's a few questions have come in about why choose RNA versus DNA. And so why?
I spent the first 6 years of my career working on DNA vaccines at Vical from 2000 to 2006, and took multiple systems into the clinic, and the data was disappointing. We saw, we were dosing milligram quantities, and as we went up through the species to larger animals and then humans, the immune responses diminished, and we had to dose larger and larger amounts of DNA. We didn't see that with RNA. And certainly, at Novartis, we started our program in 2008. There was a lot of skepticism. Okay, it's a mouse. We don't care. It'll never work in a non-human primate. Well, it did. And the dose really didn't go up. And then we didn't get it into humans, but, it is in humans with GSK. The technology transferred in 2015. We haven't seen any data yet. CureVac it's not a self-amplifying RNA, but they put a press release out that they were seeing with the conventional wild-type mRNA meaningful neutralizing antibody titers to rabies at a 1 microgram dose after 1 and 2 vaccinations. So it's not DNA. It's translating into larger species and not at high doses.
Okay, cool.
And why is that? RNA is not DNA, and our immune compartment is hot-wired to detect RNA viruses. And so that's why the wild-type systems seem to be working.
Great. And I believe we actually have a live question here from Dalit. Is that correct? So Dalit, let's hear your voice. Get someone else on here so you can ask the question to Andy and Lloyd. So go ahead, sir.
Can you hear me?
Oh, Lloyd, sorry, excuse me. Yes, we can.
Yes.
Go ahead.
Hi, how are you? Thank you for the wonderful webinar. I'm
welcome.
speaking from Israel, from Tel Aviv University.
Welcome.
I'd like to ask about cationic lipids because we need it for encapsulated nucleic acid, but it's less effective when we go to in vivo and it's so toxic. Do you have any expertise, any experience with using these cationic lipids in clinical trials? Are they safe? Can we use it? Any specific system, a platform system for inject it or use it as a vaccine?
Good question. Lloyd, maybe you can speak to that. So cationic lipids, toxic typically, obviously clinically approved lipids for the Onpattro product. So what's the difference between what we're using now and traditional cationic lipids?
Yeah, so there's been an evolution of kind of cationic lipids used for gene therapy. I think originally the first generation were these positively charged cationic lipids, permanently positively charged. Around probably kind of 20 years ago now, ionizable cation lipids started to be used for LMP formulation. So these lipids at acidic pH will have a positive charge that allow them to interact with the nucleic acid and promote encapsulation of the nucleic acid payload. Whereas when you raise the pH of the buffer environment, and in fact in the bloodstream, where it's closer to neutral, those cation lipids lose their net positive charge. Many formulations also have a PEG lipid coating as well, which shields at the surface. So in the bloodstream, those lipid nanoparticles have very little positive charge, if at all. So that really helps, allows them to be administered safely into human beings.
Yeah, I think that's a huge advance there, the ionizable cationic lipids, and I believe.
And Euan, just to build on that, the lipids that we were using and published on for our LMP formulations at Novartis for vaccine applications were those titratable amines with the pKa's in the range of 6.5. In fact, our lipid that we took and the process that we published on for making those vaccines was taken from papers published by Lloyd. So it's great to be now working with him and not just reading and copying the papers.
Yeah, and I think it's safe to say, gentlemen, that those LMP systems that are being used that we were talking about for the vaccine development of COVID-19 will be ionizable lipids. They'll be these titratable lipid systems. And so I think we're almost coming to the end. I understand we have had a lot of questions on targeting, and particularly targeting to the lung, lipid choice active targeting. I think what I would suggest is that we take some of these questions. I think we use them for subsequent webinar panel events so that we can answer them and give them the time that they require there. I think there's a question here that's maybe a good one to kind of finish on here is there any generic LMP systems that can be adapted for messenger RNA or anti-COVID drug delivery, which would save time and safety issues to help the pandemic intervention? I think that's a great question. Gentlemen, do you want to take a pop at that? Lloyd, is there a generic LMP system at this time?
No, but we need one. I'm going to maybe answer that way. You know, again, I think we're talking about pandemic response. We're talking about multiple groups, distributed manufacturing, and in many countries in the world that will help with the distribution of these vaccines. So definitely we need the one. We need standards as well for basing new development of lipid nanoparticle-based therapeutics. So, you know, standardization of a lot of the manufacturing processes and the materials and the actual formulations is really going to help drive this field forward, and is really going to make it important for the lipid nanoparticle-based vaccines or therapeutics to really, you know, make a big difference in this world.
Yeah, and I think that's a great point here. I think as we think about the future and being ready for the pandemic, so, you know, a generic or readily available LMP would obviously be one component. What other things do you think, maybe I can ask both of you, Andy first and then Lloyd, what are the couple of things that you think we could implement or right now to be more ready, if you want to say that, to rapidly respond to any future pandemic? So Andy, what do we need?
You know, so I'll just address the LMP component for the vaccines. It's got three different RNA technologies. They probably work mechanistically very differently. So we may have three different LMP solutions for those platforms, but within the platform, it's generic. Yeah. So, you know, all you're doing for, you know, for that self-amplifying RNA, and we published on this, the antigen is cross-presented. So it's expressed in the muscle cell, the immune compartment finds it and educates the immune system to be specific to that antigen. And so the delivery system can be fixed. All you're doing is varying the antigen. So you're in a position where you can build a generic manufacturing platform. You synthesize your RNA, you have a microfluidic mixing technology to form your LMPs, and then a downstream process to get it ready for your drug product and your fill finish. Now, obviously, RNA fill finish, the final format, the stability at scale needs to be worked out. But that process is generic. It's the same for all three of those RNA platforms. And I think I'll let Lloyd comment. You could probably use the same platform to produce therapeutics as well.
Yeah, so, you know, definitely. So a facility or a plant that can be repurposed and used, especially during these kind of outbreaks or pandemics, where your platform's in place, your materials are in place, you just need that missing component or the tweak to the formulation or the tweak to the payload, and you're ready to go.
That's great. So I think we've come to the end here. We've just at the bottom of the hour. So I'd just like to thank our panelists, Andy and Lloyd. Thanks for sharing your experience, your expertise with us. I'd like to thank everyone that's participated and spent your valuable time with us. If there's any other topics that you're interested in covering here, please let us know and we'll endeavour to do that. Just like you to be aware of our next webinar, which is next week on Tuesday, April 28th. That's at 11:30 a.m. Eastern Time. And this is exploring early preclinical development of mRNA therapeutics. So we'll be really examining the fundamental aspects of developing LMP formulations for gene delivery, including the process parameters and chemistry. And we'll use a model mRNA therapeutic and erythropoietin protein replacement therapy as a model drug. But I would stress that the information in this webinar will be applicable to all RNA-based therapeutics. So that's focused really on early preclinical development. So thanks very much, everyone, for your participation. Hope to see you live sometime very soon. Have a great day. Bye now.
Bye, everyone. Thank you.
Thank you.
Thanks.